And here you can find a picture of how it's possible to see what is latent. So if you look at this bag, this is semen by a bag. Okay, semen by a bag. If you look at the bag with visible light, it's impossible to see semen. But if you look with the violet in this wavelength, you will see clearly semen. Okay, also here. Another things, so this is our physical way to exaltation of biological traces, but you can use also chemical methods for exaltation of biological trace, like luminol. Okay, luminol is a chemioluminescence. So there are two reagents that react between each other and produce luminescence in presence of, for example, blood. The ring of hemoglobin in particular. Okay. So, for example, here, if you look at this is very sensible method, and you can find also blood diluted until 1 million of times. In this case, you have this was a show from a case of where an assault from two teenagers with a knife. So some blood was found on the shoes of one of them. But the important thing is that it is not possible to find by eye, but it's possible to find by using luminol as well on the knife. Okay, this is the power of this is very powerful instruments. The positive aspect, these methods is cheap, quick and easy to prepare, very sensitive, and above all, do not interfere with the subsequent assessment, with the analysis of forensic genetics. But since it is specific not for human blood, it can identify animal blood or other substances that are not blood at all. That means that you can have false positive. Also, the reaction of the luminol is very quick, it's very fast. So you will have a spot of light that can be documented by a photo. So you require some skills and experience in doing this. It's not easy. Okay. Case study 3. The victim presents itself at the emergency room with an almost completely amputated hand. Despite the injuries, he does not provide details about the attack. Why? Because he was scared. He was scared by the consequences of his testimony. Okay. Identificator identify the building where there are traces of blood, which are, however, interrupted along the internal staircase. Handkerchiefs with blood were found. A person who was already under house arrest in the building for crimes relating to international drug trafficking is being investigated. The auditorial, the judge ordered an inspection on the stairs and in the apartment of the suspect to search for blood. Okay, so we did an inspection in this scene of crime to look for what? To look for blood of the victim, to demonstrate that there was happened the crime. Okay. Of course, the suspected probably have cleaned completely the floor, but luminol is so sensitive that blood was found in the space between the floor joints. Here you can see the line, because despite cleaning completely with sodium hypochlorite, some blood remained on the joints of the floor. So blood substance accumulated in the floor joints, and it was found an unknown profile, male 1. Okay. Also on the okay, this paper folder sheet were investigated to find blood, and on this there were found 2 different unknown profile: male 1 that was the same from the floor, and male 2. Any suggestion about the contributors? Male 1 was the victim, male 2 was the perpetrator. This man was using an axe to cut the hand of the victim, but in doing this, probably he cut a part of his finger or something like that. Part of his blood was on, also probably the victim was not happy to be for this reason, and probably there was a fight. Some blood was found on this item. So then 2 unknown profile, they must be compared to someone to have an identification. The unknown man was male 1 was identified by comparison with a buccal swab taken from the victim. Unknown male 2 was identified with a suspect through a match in the national DNA databases, because the suspected man had already committed the crimes. Okay, so he was in the Italian DNA national databases. Okay, so another thing really important for forensic genetic analysis, another step is biological identification. Biological identification is made to identify the nature of the trace. So if a trace is blood, saliva, semen, vaginal fluid, epithelial cell, you can understand that having saliva from an item for a sexual crime is different than to have semen. So it's important to identify the nature of the traces. And in this case, there are many methods: enzymatic methods, chemical methods, immunological methods, histological methods. For example, for direct visualization, there are some methods to color the sperms obtained by an items, and here you have the sperm that are directly visualized, or maybe you can use other fluorescence protein to identify them, and immunological methods. Okay, this is a test exactly like the pregnancy test to say if someone is pregnant, that are based on immunological substances that recognize, for example, blood or semen or saliva. Okay, after that, so sampling, yeah, here, sampling, biological identification, then start a technical DNA workflow, which is this really technical part. I can show you a video, but maybe I can explain easily, quickly here. So basically, you have the traces. The traces is made of what? Biological cells, biological substances with the cells, DNA inside the cells. You need to extract DNA, purify the DNA from all the material. The DNA during this phase, that is DNA extraction, is isolate between the rest, the membranes of the cell, the proteins that are present in the cell. So the DNA isolated. Once that DNA is isolated, it is quantified. What does it mean? To search for how much DNA is present in the trace. And also, during quantification step, you have also some information about the quality of the DNA. For example, if the DNA is degraded. Okay, this is an important step. Then the DNA is amplified. What does it mean to amplify the DNA? It means that the original DNA is amplified. It is made a copy in a sequential way until 1 million of copy of that DNA. Not all the region, but just the marker that we were speaking about. So just that part of the DNA which interests our assessment is amplified. And after that, you will have millions of copy of the original DNA. That allows us to analyze that and have a clear results. Otherwise, it would be impossible. Amplification is done during the COVID period. Your DNA, to find the DNA of the virus, was amplified to detect the virus during the COVID. You remember the test that was done in that period? The DNA was amplified to be possible to be detected. Okay, so similarly, the DNA from the trace is amplified to be possible to be correctly detected. The entire workflow is recorded on a LIMS, Laboratory Information Management System, which is a software basically that records all the activities conducted on the traces, on the samples. This is important for the accreditation according the ISO IEC 17025 that I showed at the beginning of our discussion. The last step, of course, is the interpretation of results and the reporting to the court. Okay. So, for example, here you have an example of the reporting to the prosecutor. And interpretation is not usually so easy. Before I show with you a single profile that is here, very clear. No? You have the DNA of just one person. You can find the characteristic for each marker, okay? Many markers. But what happens if the DNA is not enough? You will miss some signal. So this is a low template, is called, this kind of DNA, in which the DNA is not enough and the results are not so clear. Look, completely different. You have some dropout, are called, but basically you lost the signal. Okay, or you can have artifact. So the interpretation becomes complex, really complex. Another point is a degraded DNA in which the DNA is cut basically, and so you will not have the biggest part of the DNA. You will have only small part of DNA, and then also some mixture profiles. This is a mixture profile from at least three contributors, in which in pink you have the presence of a victim, for example. It's not always possible to understand if a contributor is present or not. It's not so easy. Interpretation can be really difficult. Okay, If you have a result, a good result, maybe not this, or maybe not this, okay? If you have a good results, you must do a comparison. As I told you, the comparison can happens with known subjects, suspect, crime victims, because you want to identify all the unknown profile, or with the DNA database, but also with family to investigate a kinship relationship. Okay, this is particularly important for human remains and missing person casework. So, just few words about the Italian DNA National Database. I told you that the law is 2009, but the database was started in 2016 or 17. Okay. Basically, the database is done by two macro database. One database is done is where are uploaded all the unknown profile that arrive from the crime scene, the scene of crime. All the profile are in this database, and then another database is from a reference sample. So the profile, genetic profile that are obtained by convicted or arrested fellows. There is a specific article of the law that say that some people are the DNA are taken from some people that are in arrested or convicted condition. These two databases are searched one. So when a new trace or a new reference sample is uploaded, it is searched against the other database. All the information are unknown, are decodified, well, are codified, and the only person that has all the entire clear view of the aspect is the judicial authority. Because, for example, when we have a match, we don't know the name of the person that has produced that match. We know only that the trace is obtained by a scene of crime has matched with a person, unknown person, just a code. The person who has the name of the trace and the name of the crime scene and the name of the person is the judicial authority. This is because of the privacy. This is because of guarantee for all the person involved. Okay, we are at the end. At the end, well, just speak about, I showed you that the technical part of the DNA analysis is complex, made of many phases, but actually nowadays well, there are a few instruments that have compressed the entire workflow. They are called the sample in, profile out devices that allows the analysis in 19 minutes. This device is important, can be really important for many aspects, but one of these is an investigative lead. Consider that if you have many, I don't know, a crime of scene, a scene of crime, and you want to have an answer quickly, and you can use this kind of device. This kind of device is currently used not in Italy, but in America because of their legislation. The legislation of FBI for DNA uploading on the US national database allow the use of this instrument. In Italy is not really possible, so we have a different legislation, but it represents an aspect of the future. This is this instrument is easy to use, is portable, of course, fast result for fast investigative leads, and also reduce the vast. Another aspect that we're speaking about before was the DNA phenotyping. Currently, it's possible to have some information by biological traces, especially for investigative purpose. At least the technology is ready for this, and as I told you, for in certain countries is allowed for by the law to do this kind of investigation. And it's possible to have some information about the color of the skin, the color of the eyes, and the color of hairs. This is important for what? Because if you don't have any clues about, if you don't have a crime, if you don't have a suspect, well, maybe this can help you to reduce the number of suspects. Maybe if you have the color of the skin, you will have some information of the color of the eyes, for example, can help you to remove some suspect from your list. Okay, this is another powerful, really important. So sorry, another instrument really powerful. But what about DNA typing in a CBRN event? The problem of a CBRN event and genetic investigation is that you can use forensic genetics for all the crime scene analysis if you don't have contamination by CBRN materials. Because if you have an item that is contaminated by chemical, by biological substances that are CBRN substance, well, then you need to decontaminate, and the protocols for decontamination currently, in the most of case, are the extractive way that will destruct also your DNA. So this is basically an aspect that should be considered by when thinking about forensic genetic investigation and CBRN event. But despite this fact, for example, if you don't have presence of CBRN substances, well, you can do all the things I've said to you. I mean, the case of the fake IED, the device, is a case happens in a terroristic attack, for example. Okay, the case of Luminol as well. So this aspect can be considered, and also the contamination phase must be considered in case of CBRN event. Usually in the lab we receive some envelope in the treat case, when I don't know, some let me see there. Some envelope of mail are sent to someone to treat him. Okay, in that case, sometimes you have, I don't know, powder, maybe, or with substances there, maybe writing as anthrax, but sometimes they are not anthrax, of course. But in that case, anyway, before to be analyzed, they must be decontaminated. Decontamination, in that case, decontamination of biological substances like anthrax spore consists in treatment at least for 20 minutes at 120 degrees. Okay. This destroys spore. It can destroy spore, or the use of for chemicals. This destroys spore, but destroy, of course, DNA of the perpetrator. So this must be considered. Okay. Many thanks for your attention. I don't know if there are some questions. No? No one survived. Okay. I think we can have 5 minutes of break, and then we can have a short demonstration of some fingerprints, maybe something also for biological traces. Ok, thank you. Okay. Allestiamo? Ok, sì, facciamo il nostro meglio, a prescindere.
Stefano, scusami, la parte di slide è finita?
Scusa, sì, la parte delle slide è finita.
Ok, quindi fermo la registrazione perché non abbiamo più altro da registrare,
Ok,
giusto?
va bene, sì, sì, assolutamente.
Ok, grazie.
Ciao Colomba, ciao.